Hi students, good morning, welcome again. Today I am again revising the chapters of class 10 ICSE, the important points only, starting with chromosomes and cell cycle and cell division. The chapter cell I am leaving because number of times you have studied that chapter, and today I am starting with chromosomes. So structure of chromosomes, coiled chromatin fibers, composition, DNA and histones, that is proteins, and DNA 40%, histones 60%. Now DNA structure, complementary two strands in double helical structure. Each strand, segments are there, nucleotides, and each nucleotide is composed of your a sugar attached with phosphate and a nitrogenous base, which is again linked with another nitrogenous base of the another strand. What are the nitrogenous bases here? The adenine, thymine, your guanine, cytosine. Adenine will go with thymine, thymine with adenine, cytosine with guanine, guanine with cytosine. The bonds are there, hydrogen bonds, and DNA when is around, DNA strands when they are around the 8 histones, that is proteins, then it is the unit formed is the nucleosome. So these things you have to remember. And in the chromosome there is a segment which is the gene which is carrying the similar characters from parents to offsprings, and that is why all the members of a species, all the organisms are similar in looks, not identical but similar, because the similar characters of the parents are being transmitted to the offsprings by the genes. Okay, so this is a segment of the nucleotide. Gene is a segment of the nucleotide which is having the characters coded, and these characters are appearing in the offsprings. Okay. Now the cell cycle. Every cell has to go to the has to enter the cell cycle. Most of the cells in plants as well as in animals. Okay. So the cell cycle is in 3 steps, as you all remember. The G1, the synthesis phase, and G2. G1 means growth phase, growth 1 phase, where synthesis of protein and carbohydrates will be there. These points only I want to just to revise so that you, as a ready reference, always you can remember them when you hear this video. This is my intention. You have to study the chapters thoroughly, otherwise this will not help. That is true. But what I am trying to do is, I am just in a nutshell, I am putting the important points in the same video of the chapters so that it will help you in remembering, okay, in revising, right? So this is the cell cycle I am talking about. G1, synthesis of protein, synthesis of DNA and RNA will be there. Mitochondria, chloroplasts will divide because they have their own DNA. Some groups will enter, of cells will enter the cell cycle. Some will go to the resting phase. Now, this is G1. Then synthesis phase will be there, which will, in this phase, maximum DNA will be synthesized, okay, and the chromosomes will duplicate. In the G2 phase, all these formation of DNA, RNA will continue. Fine. This will be a shorter phase. Now, cell cycle will be there, but in all the parts, in all the organs. But continuation of cell cycle, continuation of cell growth is also not, expected, because that can lead to some tumors. So the number of cell cycles will vary. In the brain, it is only once. It cannot be changed. It cannot be renewed. In the liver, it is once in 1 or 2 years. In the skin, is always, the rapid changes, rapid. This thing is renewal of the cells are there in the skin. In the plants, these meristematic tissues keep on dividing up till some time. So like this it goes on. Now cell division, in the cell division is the general cells, so they are dividing. So what are the stages there in the cell division? That is the mitosis and meiosis. For reproductive cells it is meiosis, for general cells it is mitosis. Mitosis have two phases. One is karyokinesis, that all the changes in the nucleus is taking place, and the cytokinesis, where the cytoplasm is divided into two. So that is mitosis. That is how new cells are developed. And in this in this mitosis, the phases are prophase, metaphase, anaphase, telophase. Idea is that the first of all, the primary stage, the nuclear membrane will not be there, will be there, but it will start forming, and chromatin fibers will be there, very very faded like picture will be there, and they will gradually become distinct. The chromosomes will become distinct, nuclear membrane will appear. So this is the prophase, and then in the metaphase they will become chromatids, and later on chromosomes. They will be joined with a centromere, and there will be there a pair of chromatids will be there in the center. Okay, and in the metaphase they will come into the central line. In the metaphase, what is happening? They are coming into the central line. Okay, in the equatorial line. And asters have been formed in animal cells, because centrosome was there in the animal cell, which is not there in the plant cell. So plant cell will not have asters, but spindles will be there in both the cells, animal and plant cells. So these spindles will be attached to the centromeres of the pair of the chromatids in metaphase, and then in anaphase, these chromatids, both the parts of the chromatids, both the hands will get separated, and they will move towards the pole in anaphase. So the asters will there, and they will gradually move to the members of these pairs will move, each member in each side, in each pole, they will move towards the poles, and spindles will be there. In telophase, what is there is a cleavage furrow will appear. It will deepen down the animal cell and start dividing, and two nuclei will be formed. And this nuclear membrane will again disappear, and these chromatids will again go back to that form, that is chromatin form, the very very thin threads. So the stage where it has started, and in this ends the phase of karyokinesis, and then cytokinesis starts, and this cleavage furrow deepens down and divides the cytoplasm into two daughter cells. And plant cell, the plate is there, which is dividing the cytoplasm, and it divides into two daughter cells. So this is mitosis. And in meiosis, it is for the gametes only, for the reproductive cells. In the first step, it is a reduction division. Diploid chromosome will become half, that is haploid, and again mitosis will take place, and then 4 individual chromosomes of cells will be, independent cells will be formed. So this is in short the meiosis. Okay, I don't think a detail of meiosis is required, and difference is between mitosis and meiosis is already you have understood. Mitosis is for general cells, meiosis is for reproductive cells, and mitosis is for growth and replacement. And why division takes place? Cell division takes place for growth, number one. Number two, for damage repair. If any cell is damaged, it is repaired. Okay. And division is for reproduction also. Cell division is very very important. So that is the significance of cell division. Mitosis and meiosis is especially for reproductive cells. Okay, then we come to the basic principles of genetics. Here, children, you have to remember that Gregor Mendel was the father of genetics, who said that how we look similar and why we are not identical, why there is always a difference, little difference. So that is the heredity, principle of heredity, which explains this, and also variation. So he explained all these two aspects of genetics, that is heredity and variation, and he also defined that characters and traits are different. Character is that color of the eye, it is blue or red, blue or white, blue or black, it is the trait, and then character is the color of the skin, it is fair or not fair, it is the trait, or color of the vision, okay, normal or red green color, it is also the trait. So like this he defined characters and traits, okay, and there is a variation in population also. So why this variation takes place? This is because of the when duplication of DNA takes place, it is not always completely correct. Some errors are there, and some mutation also take place because of the environmental change, because of the internal and external changes, and that and for that also variations take place. So in a family also looks are different, so that is because of the variations. So an individual feature of organism is a character. The alternative forms of a character is called the traits. Okay. So this he tried to define, and chromosome, number of chromosomes is fixed in organisms. We have 23 pairs, and autosomes and heterosomes. Autosomes means similar. A pair of chromosomes already always be there. So it is X and Y. Y means what? There is a 23 pairs of chromosomes we are having. 22 pairs are similar. They are autosomes, and one pair is sex chromosomes, where X and Y, two types of chromosomes are there, which are dissimilar chromosomes. So X chromosomes means what? XX means they are both the autosomes are there, so they are the female, chromosomes in the female. In the male, it will one dissimilar chromosome will be there, that is the Y chromosome. So it is XY. So in male, it is always both the chromosomes will be there, X and Y. But in female, it will always be similar autosomes, similar chromosomes, that is X and X. So the determination of male or female child, sex determination, is always depends on the male's chromosome. The male is having X and Y, both the chromosomes. So if the X of male combines with X of female, the female is having both the X chromosomes. So if X of male is combining with X of female, then it will be XX, that is girl child. And if Y of the male is combining with X of the female, then it will be XY, so it will be a male child. So determination of sex in human is dependent on the father, not on the mother. So in the male chromosome. So it is always to be clear that how sex determination is done. So these questions come generally about the sex determination, I think it is clear. And genotype and phenotype, you have to remember, genotype means the genetic constitution, the constitution of the genes, and phenotype means how the characters are appearing. So the dominant characters are shown as the capital R, and of the same letter, if it is denoted by small letter, then it is recessive trait. Okay, so RR means both dominant. Always it will be in pairs. The genes will be in pairs. Okay, small RR means recessive character. Okay, dominant and recessive you have to understand, and the laws that he derived from his experiments. So pea plant he took for his experiments because they are in abundance. They are having many varieties. They are a short span of life, that is seasonal flowers. So it was very easy for him to see the results of every generation. So it was very convenient for Mendel to study with pea plants. Okay, and these are the things which are important in this chapter. Okay, and the laws that he derived by his experiments were that the Mendel's first law was what was that the in a monohybrid cross, the cross the result will be in F1 generation. If we take a monohybrid cross of contrasting characters, say red flower and white flower, then what will happen in F1 generation? The one color in this example, red is if it is dominant, then all the flowers will be red. All the four flowers will be red in the F1 generation. See, when there is a cross being made, then both the genes will be there in each offspring, in each flower. So, but the red is dominant, so red is appearing, and the white color is recessive, so it is hidden. But in his second law, he said that the hidden character also segregates itself. So, in this case, the white color will segregate in the ratio of 3:1. So, 3 flowers will be red and 1 will be white. So, this is in a monohybrid cross. The first law is the law of dominance, and the second law is the law of segregation. And the third law is law of independent assortment, that is in a dihybrid cross. Okay, in a dihybrid cross, there are two pairs of contrasting characters are taken, and there what he observed is that all the characters are being shared by each pairs. It is independent of the other pairs. So this is independent assortment. All the characters will be there in the combinations. So for example, round yellow and wrinkled green, if you cross, there's two pairs of characters. One is of the shape and one is of the color. So the dominant here is round, round yellow. So round yellow will be 9, round green will be 3, wrinkled yellow will be 3, and wrinkled green will be 1. So all the characters have appeared. So in the ratio of 9:3:3:1. Phenotype. Their genotype is very difficult to derive at. So genotype you need not study, and need not study or know at this stage. And the first law, that is law of dominance, is the phenotype. What we see is 3:1, and the genotype ratio is 1:2:1. Okay. The law of dominance is all the dominant characters, but it is the second law, that is this recessive character appearing in the ratio of 3:1. Okay, so first law, there is no ratio. First law is the law of dominance. So all the flowers are red. So the red color is dominant here. But in the second, the recessive has segregated itself, the recessive character, in the ratio of 3:1. That is phenotype. And genotype here is 1:2:1. Okay. So these ratios you have to remember, student. In the monohybrid ratio, I am again repeating, in the monohybrid ratio, the phenotype ratio is 3:1, and genotypic ratio is 1:2:1. And in dihybrid ratio, phenotypic ratio is 9:3:3:1. And genotypic ratio, as I said, is very complex. And about mutation. Mutation is a sudden change in one or more genes, or in number or in structure of chromosomes. So this you have to remember. Mutation alters the hereditary material of an organism. So sickle cell anemia can appear because of mutation. It's a blood disease caused by a gene mutation. The mutation causes changes in DNA, resulting in the production of sickle-shaped RBCs. So sickle cell anemia can be a result of mutation. Radioactive radiations also after the gene structure and their effects can be seen generation after generation. An atomic explosion which had occurred during World War II is an example of radioactive mutations or radiations. Okay. Other minor points when you go through, you can easily revise. Absorption of roots is the chapter, and the processes are what? Main processes are osmosis, diffusion, okay, and your active transport. Osmosis means what? Water from its higher concentration moving to its lower concentration is osmosis through a semi-permanent membrane. Diffusion, just recollect, is the same. Water from its higher concentration to lower concentration without any membrane. Any substance from higher concentration to lower concentration without any membrane. Diffusion is mainly in gases. And then the active transport, bigger molecules entering the cells of the plant, without any membrane, but from lower concentration to their higher concentration, and the energy used is obtained from the cell only. So this is active transport. And then the other things which are there in this chapter are the imbibition, which is what? Which is the surface attraction. The living or dead plant cells absorb water by surface attraction. This is imbibition, and turgidity, water is pushing hard against the cell wall of the plant cell. So that is the wall pressure, and that is because of the turgidity. And flaccidity, when the plant cell is releasing water, when the water is getting out of the cell, that is exoosmosis is taking place. So that is flaccidity. That condition is called flaccidity. And your isotonic and hypotonic, hypertonic. Hypotonic means endoosmosis. When endoosmosis is taking place, it is hypotonic. When the water is coming into the cell, it is endoosmosis. When water will come into the cells? When water will, from the outside, from its higher concentration, comes into the in its lower concentration inside the cell. That means outside, what is there? Water is a higher concentration, solute is in low concentration, so solution is having a lower concentration. Okay, so solute is low concentration and the water is in higher concentration, so water is entering the cell due to osmosis, that is endosmosis. And if the water is coming out of the cell, because in the cell water is more, that is the solution is diluted, so water concentration is high, so it is coming out of the cell, so it is exoosmosis. So it is a hypertonic solution. In endoosmosis it is a hypotonic solution, and in exoosmosis it is a hypertonic solution inside the cell. Okay. So this you should understand very clearly. In hypotonic solution, what is happening? In hypotonic solution, the solution outside the cell has a lower solute concentration than the fluids inside. That is, water is entering inside. Why? Because water outside is in higher concentration. Okay, solute is in lower concentration outside, but the water is higher. That is why water is entering inside the cell. So the situation is, hypotonic. As a result, the water molecules from outside will move into the cell. That is, endoosmosis is taking place. In this condition, the solution outside the cell has a lower solute concentration than the fluids inside the cell. So, the concentration of the solution outside is low. So, it is hypotonic. If the concentration is low, the water is more, the water will enter. And in hypertonic, outside the solution concentration is high. In hypertonic, solution concentration is high means water is less. So, water from inside will come outside because water is more inside. So, it is coming outside. So it is a hypertonic solution outside. Hypertonic means the concentration is high. That means solute is more, water is less. So if water is less outside, so that means water is more inside the cell. So from its higher concentration, water will come out through the cell membrane. So this is the hypertonic solution outside. This you have to remember. If the solution concentration is same on both the sides, then it is isotonic. Okay, so these are the figures which you have to just practice and revise so that you don't miss whenever they come in the exams. Transpiration, you all know is what is transpiration? Transpiration is loss of water through the stomata. Stomata opens and closes because of the guard cells. Turbidity of the guard cells will open the will make the opening wider, because when water is filled there will be a turgor pressure which will in the guard cells, and the opening will be wider, and when the water has gone out, that is exoosmosis has taken place, the cell will shrink. Okay, so that is flaccid condition. So what is because of this, the transpiration is taking place, water vapor is released out of the stomata, and this water loss is creating a huge force in the columns, that is the transpiration pull, and because of this water and minerals are pulled upwards in the xylem. So transpiration is so important. It is creating a pull which is responsible for driving the water minerals upwards in the xylem. And what are the demonstration of transpiration experiments? That is there in the books. You just go through them. And how this rate is measured? The transpiration rate is not measured, but the rate of uptake of water in the photometers is seen, and the most important or common experiment done is with the Ganong's photometer. So it will not measure the water lost during transpiration, but measures the water uptake by the cut shoot. Okay, the bubble will, when we enter make the bubble enter the curved tube, then what will happen? The bubble will move forward in the tube when water is lost through the leaves. So we can see that how much uptake has been done. Uptake by the cut shoot, how much water has gone in, that we can measure. So we cannot measure the water, how much water has been lost. What is the precaution in the use of photometer? The photometer should be made completely watertight. The twig should be cut obliquely to allow larger surface of water intake. So these are the points which you have to remember, students, in this chapter of transpiration. What are the factors which can be there, which can affect the transpiration? That is, the rate of transpiration will go higher in case of higher temperature, in case of broad sunlight. In case of low temperature, the rate of transpiration will go down. The humidity, if it is more in the atmosphere, the rate will go down because the vapors will not have enough space to be accommodated in the atmosphere, so the rate will go down. Okay, these are the factors which will affect the rate of transpiration. And kinds of transpiration can take place from the stomata, which is the main route, that is the stomatal transpiration can take place through the cuticle surface of the leaves directly, cuticular transpiration, and also from the lenticels on the stems, small pores. So that is lenticular transpiration. So transpiration can be stomatal transpiration, can be cuticular transpiration, and can also be lenticular transpiration. So external factors, internal factors of transpiration you have to remember, which can have an impact or affect transpiration. As I just now said, intensity of sunlight, temperature, velocity of wind also can matter, because wind will move the vapors quickly, and so more and more transpiration can take place. Carbon dioxide increase in the CO2 level in the outside air over normal causes stomatal closure. Stomata will close, the transpiration rate will go down. Internal factors are there. Some reduction in transpiration is indirectly due to the closure of stomata. So there can be your sunken stomata, there can be narrow leaves, there can be thin leaves, and loss of leaves can be also there. So if the leaves are not there, stomata is not there, if stomata is and if the leaves are narrow, stomata are less in number, fewer in number, so all these will have an effect on the rate of transpiration. Cuticle is thick, water vapor will be the leaves may be covered by thick cuticle, so it will be difficult for these leaves like banana, banyan, and most evergreen trees. Thick cuticle they are having, so transpiration rate will slow down. Significance of transpiration I discussed already. Cooling effect it does. It creates a suction force, and which is pulling the water mineral upwards in the xylem. So like this, you have to remember the salient features of transpiration, students. Photosynthesis also, you all are aware of photosynthesis. You just go through the light phase and dark phase once again. Light phase is the phase where light is absorbed by the chlorophyll, and then chlorophyll is activated, and then photolysis takes place. Water is broken down into hydrogen and oxygen, and some electrons are also being released. So what is happening? Hydrogen is being released, and it is combining with a compound NADP, which is making NADPH. Okay. And then this oxygen, free oxygen we are getting, molecular oxygen, and then these electrons released are converting the ADP to ATP, adenosine diphosphate to adenosine triphosphate. So this is the phosphorylation which is taking place, which is called the phosphorylation. So this is very important in the light phase, because this will not take place without light. Okay. Activation of chlorophyll and splitting of water. These two steps are taking place in the light phase, which is very, very important. And NADP is what? Nicotinamide adenine dinucleotide phosphate. So this compound is combining with hydrogen, which is again released in the dark phase, and carbohydrate is formed because of the reduction of carbon dioxide which is present, which is coming through the stomata. So this is the light phase, and in the dark phase all these assembly of these matters which we have got from the light phase will take place. That is, oxygen is there, hydrogen is there in the free state from NADPH, it will be released, and then carbon is there after the reduction of carbon dioxide. So carbohydrate, that is C6H12O6, will be formed. So dark phase, light phase, dark phase. Dark phase is light independent phase. For this to happen, light is not required. Another thing happens in dark phase, that is the starch is formed. This, the conversion of glucose into starch. This is take place, and other chemicals. This will take place in the dark phase. So these things you have to remember, students, and adaptation of leaf for better photosynthesis, large surface area, leaf arrangement. All these, if you understand, the light will be more. If the light is more, the photosynthesis will be better. So all these adaptations are to get maximum light. Leaf arrangement, cuticle and upper epidermis, numerous stomata, the thinness of leaves, okay, etc. The chloroplast, if the chloroplast are concentrated at the upper part of the leaves, then chlorophyll will be more there at the upper part, and the more light will be absorbed. So extensive vein system, if it is there for rapid transport to form the mesophyll to and from the mesophyll cells. So the transportation, if it is faster, photosynthesis will also be faster. Water has to go, carbon dioxide has to go, and then finally glucose will be formed. Factors affecting photosynthesis: naturally light intensity, availability of carbon dioxide and temperature, water current, water content in the soil. So these things are very, very easy to understand if you are thorough in the chapter. I am just discussing the important aspects. What is the importance of photosynthesis? One, it is providing food to the plants and also to all the organisms, directly or indirectly. In the food chain, you are saying the producer is plant and the consumers are all animals, including man, but they are indirectly dependent on plants only, on the producers. So it is providing food. Secondly, the natural biggest source of oxygen is photosynthesis only. We are taking oxygen and we are releasing out carbon dioxide, which is maintaining the balance in the atmosphere. So to maintain the balance in the atmosphere of oxygen and carbon dioxide, the trees, the photosynthesis in the trees are very, very important. Then the carbon cycle and food chain already I have discussed, and respiration, decay, this combustion, heating, limestone. So how carbon cycle is completed, how carbon dioxide is there, the balance is maintained. So here some certain points you have to remember, students. By photosynthesis, by food chains, by respiration, all plants and animals respire by oxidizing carbohydrates in their cells to produce energy and give out carbon dioxide into the atmosphere. We already talked about this. So I think the other chapters left we can do in the next video before it becomes too long. I think it will help you, and thank you very much, and wish you the very best. Thank you.